Thank you, chairpersons. And also at the outset, I thank the organizers, especially Dr. Nag and Dr. Gopala, for inviting me to this particular meeting, which is very unique in a sense that we have been discussing that where there is a confluence of the clinicians and the basic scientists, that we are discussing the various aspects of cancer research and its implication in its management. What I'm going to discuss today is a small story that has been unfolding in my laboratory over the last couple of years. It's for the first time I am talking about this story in a public forum, which we are very excited of and really got some interesting results that I'm going to share with you. My laboratory is basically interested to understand one of the most fascinating problem in cancer biology, as you see in this slide, where you see in the cancer cells a large number of chromosome complements are present compared to the normal cells. We are interested that how you can see that there are various kinds of chromosomal abnormalities are there in these cancer cells, starting from chromosomal structure alterations as well as the numerical alterations. And we are interested in to understand why in a cancer cell there are numerical alterations happens. There are various reasons. Due to constraint of time, I cannot talk all of them, but I will just focus on one particular aspect of this mechanisms by which in the normal cell this chromosome number is controlled or regulated so that it ensures during the mitotic division that equal number of chromosomes are distributed or segregated into the daughter cells. As you know, during mitosis, the spindle fibers from both the poles emanate and they attach to the kinetochore of the of these sister chromatids. Unless and until this kinetochore is occupied, all the kinetochores are occupied by the sister chromatids, the anaphase doesn't initiate. And in order to ensure that it doesn't initiate before the occupancy of this kinetochore, there is a checkpoint mechanisms which ensures, monitors that all the kinetochores are occupied. And in that checkpoint, the molecular molecules that involved in the checkpoint are the BAB-MAT group of proteins, which sitting on this kinetochore sequesters an adapter molecule CDC20, which actually required for the activation of the anaphase protein complex, which is the E3 ligase. And if this APC is actually required for the degradation of securin, which holds back this particular separase protease called separase, which is required for the cleavage of the cohesin molecules that holds together the two sister chromatids. Unless and until all the chromatids are attached, this is inhibited and the separase cannot activate, so anaphase doesn't initiate. As you can see more closely in here, that on this particular kinetochore, actually there is a complex of MAD1 and MAD2 protein which acts as a template where this MCC complex is formed and inhibits the CD20 molecule to activate the APC. Today, what I'm going to talk about is mostly revolved around this MAD1 protein, which is very crucial for the formation of this MCC complex. As you can see in this slide, that if for some reason this weight anaphase signal is not properly executed, then an anaphase initiates without attachment of the spindle, then what happens is that the pair of chromatids pull to the same toward the same daughter cells instead of going to the opposite daughter cells. As a result, you generate aneuploidy. And this is one of the mechanisms by which aneuploidy happens, and people want to know that it suggests that if there is a defect in this checkpoint, then this thing can happen, and wanted to know how these checkpoint defects are happening in the cancer cells. So what appears that there is a deregulation of the SAC proteins, which actually results in defective SAC, and that results in the chromosome instability. Now, there are people try to look for the mutations in the various genes of the SAC components, as well as various studied the altered expression of the SAC components, and what we have been found that in most of the cases, there is very rarely mutations have been observed in these SAC genes. Instead, there is an altered expression of these checkpoint proteins, which is actually more common and is thought to be more responsible for this defect. Actually, this has been supported by a large number of mouse models, various kinds of transgenic and knockout knock-in mouse, where in the most of the cases, actually the mouse alleles are either overexpression of the corresponding gene or the hypomorphic or the haploinsufficiency that results both this chromosomal instability phenotype, and either it generates the tumor formation in the mouse, or this slowed down the tumor formation in the mouse. In fact, actually, we just got recently a very interesting data, which I am just going to share with you. We are together with studying the cancer. We are also studying the normal meiosis in the human, using the model where in abortions. As you know, there are certain females who undergoes miscarriage quite frequently. So we are wanted to know why they are happy, what if there is any defect in these checkpoint genes in those patients. So we collaborate with a hospital in the Pune, which give us the fetal cells, primary cells, and we study the expression of these genes in those cells. And to our surprise, while studying the 20 aborted fetuses, we observed in 3 cases there is a deletion in the exon 5 region of one of the critical SAC gene, the MAT2, and interestingly, this deletion is transmitting as in a Mendelian fashion, suggesting there is this deletion is inherited by the human population and could be transmitted in inherited manner which predisposing the individual for frequent abortion. So, seeing all these things, it is being clear that rather than complete loss or what is happening in the cancer is that there is either upregulation or downregulation of these genes happens, and that results in a kind of a similar phenotype of tumorigenesis and chromosomal instability. So it appears that in these genes are not totally overtly changed, but rather there is a fine balance of these gene products are necessary to maintain the proper checkpoint function as well as the accurate chromosome segregation. So this actually, as have been proposed earlier, as you can see, actually what we now people believe that the genetic instability, which is one of the component of the hallmark, as shown in the morning slides, so that genetic instability, it has to be optimum to really go drive towards the carcinogenic pathway. If it is too much instability, it might also harmful for even the cancer cell, and cell will die. And I will show you my data suggests that this can be a therapeutic options for cancer treatment. In our laboratory, we are studying various aspects of this regulation of these expressions of these SAC genes and their proteins.